Good morning, everyone. It's a great pleasure being here. Really an honor speaking to you all to so much of the history of BTI. I was hired in 2007 as part of a move by BTI to place more emphasis on chemistry. And what I want to tell you today is, why does that make sense? Right? Why do we need to go back and look at chemistry? Isn't the chemistry of life well known? Right? There are these large molecules that Lucas was talking about, the chromosomes, that have the DNA, and that we can characterize, as Lucas just told us, in a few weeks. Right? We can get a full genome from a plant, from any human individual, very, very fast. And then there's the proteome, which is derived from the genome. And those, again, are very large molecules. And again, they're fairly straightforward to characterize. We can get a full proteome from any sample, perhaps in a few days. Thousands of proteins annotated. So those large molecules are fairly simple now to characterize, and that's due to the fact that their chemistry is inherently simple and predictable. So in DNA, or RNA, we have the same building blocks every time. Right? Let's see if I get this laser pointer work. Yes, there we go. Right? So it's the same building blocks that are connected with each other by the exact same chemistry again and again. So you can build easily, well, it took a couple decades, right? But you can build platforms that give us the entire sequence of a chromosome, perhaps. Which is still a challenge today, right? Get really a full chromosome with all the different regions in between. Okay, those are the large molecules. But that's not the entire inventory of the chemistry of life. What's missing is the vast space of small molecules down here, which I have kind of disappearing in the dark because it's really dark matter. The human body contains likely more than 100,000 compounds that are unknown. We know perhaps 5,000 compounds, right? So 5% of the chemicals in the human body, and for most organisms the situation is way worse, are known. So next time you go through airport security, you just got to say, I'm bringing with me 95,000 of unknown chemicals. And they'll be very pleased. Okay, so good. So that situation is the same for humans, mouse, Arabidopsis, everywhere. Okay, so we need to tackle it because the central hypothesis that motivates us, my lab, us chemists, is that most of these molecules are the products of dedicated biosynthetic pathways that have a function. And so they're essential for the organism to live and for it to interact with other organisms. And then what we believe is that this process of discovery, of annotating the chemical structures, finding biosynthetic pathways, finding the targets, will really help us revolutionize how we do agriculture and how we approach the human condition, how we deal with disease. And I will give you two examples for that today where that seems to be working. So in this slide, I just want you to appreciate how complex and daunting the task is. So it's a picture of the human body, and note that here we have enlargements of certain areas highlighting the presence of microbes. So the human body consists of several hundred different tissues, each tissue having different cell types, each cell type having a different metabolome, meaning the specific small molecules, the specific chemicals each cell makes are different and not predictable, right? That's the point I have to make. The DNA, you can always say, yeah, it's the same building blocks, they're connected in the same way. With chemicals, that's not the case. So I have this vast diversity of different cell types of the host, and then in addition to that, it's the microbiome. The human body contains vastly more bacterial cells than human cells, and they belong to many different species from different phyla that all contribute to the chemistry, and their chemistry interacts with the host chemistry. So it's the back and forth. That's where this complexity arises that has really prevented a systematic annotation. So when I started my lab here at BTI a good 15 years ago, we thought we need a simple model. And really the most chemist-friendly model. And also the model, I think, with the greatest potential for studying ecological interactions on a really broad scale is the nematode C. elegans. Small soil worms, they're harmless. Their relatives are parasitic. That's parasitic worms of plants and animals. And that is part of their appeal. So C. elegans, even though they're little worms, they have high homology with what's going on in a larger animal. They have different cell types, but they're extremely easy to grow and they have a very short generation time. And most importantly, we can edit their genome in a manner that's impossible for any other eukaryote. You can induce a gene at any time point. You can knock out almost any gene in this organism without creating a lethal scenario. You have tools available here that allow you to very rapidly screen thousands of mutants. So this is the basis that we've been using to investigate the role of small molecules produced by C. elegans and produced by C. elegans and other nematodes as they interact with bacteria and as they interact with mammals, higher animals in general, and with plants. Okay, so first example that I'll give you is about interaction with higher animals. And the basis for the specific study here that I'm highlighting here is a vast body of evidence that infection with parasitic nematodes can prevent or reduce the effects of autoimmune disease. Now, autoimmune disease is a huge problem in Europe, in my family as well, in North America, and much less so in Africa and in countries with more parasitic nematodes, and that was the cue. So it seemed that somehow parasitic nematode infections, even transient ones, make the body or reprogram higher animals to be less likely to suffer from autoimmune disease. And many people have been looking at proteins secreted by nematodes, but we thought that perhaps a specific type of chemical, shown here with one example, these ascaricides, which are produced by all nematodes. They're produced by C. elegans, a model system, but they're also produced by the parasitic nematodes. That this specific compound could perhaps be a signature that animals have evolved to recognize and then adapt their immune response to. And that was the basis for a large number of experiments that we did in different autoimmune disease models. And I'm just showing you one example here in colitis. And in all of the model systems that we tested, includes also asthma and type 1 diabetes, what we found is that exposure to very, very small amounts, really trace quantities of these ascaricides, prevents the development of autoimmune disease. So for what we're looking at here is weight loss due to colitis, induced colitis in mouse. And if you treat just with a tiny bit of ascaricide, in addition to trying to induce colitis, you end up with no weight loss and a healthy mouse. You can see here is a colitis colon, and here is a colon also under colitis conditions, but with a little bit of ascaricide. So, as I said, we already showed this and published it for asthma, and it also works in type 1 diabetes. So that led us to recognize that perhaps that's the potential, really, to approach treatment of autoimmune disease from a different angle, and that is the basis of one of the startups that have come from my lab, Holoclara. Holoclara is based in California, but we're very, very heavily involved with further development of this compound. Now, back to BTI. What BTI really made a great home for my lab is that us chemists could interact with biologists. And there's a great tradition here at BTI to discuss science at the coffee machine. And so when Dan Classic and I met, maybe in 2014, at the coffee machine, and I told them about our results in mice, that compounds, these ascaroside compounds from nematodes, were perceived by mice as a nematode-specific signature. Dan immediately said, oh, we have to test this in plants, right? We have to check whether perhaps plants also have evolved the ability to recognize these compounds. And in fact, Dan was right. A slightly different variant, shown in red up here, this compound has dramatic effects on plants, on monocots, on dicots, doesn't matter whether it's rice or wheat or tomato or soybean. In all of these plants, exposure to just trace amounts of this compound primes the plants to become more resistant to oomycete, fungal, bacterial, and viral infection. And the effects are very dramatic. In this case, we have soybean plants. These soybean plants here, they were just these soybeans. They were planted, they were allowed to grow, and then infected with an oomycete pathogen. These plants are from seeds that were briefly submerged in a solution of this compound. So it's a seed treatment with just a little bit of this compound that primes the plant to be more resistant to pathogen infection. And we have seen that this is really priming at the DNA level, right? It changes in the accessibility of the genome in response to treatment of very small amounts of this compound. So that's the basis for the second startup that has come out of my lab, Ascribe. Ascribe is incubated here at BTI, so you can actually see some of the Ascribe labs here on the first floor, and BTI has been really tremendously supportive to get this startup on the way. We now have field trials all over the planet, in Australia and India. We just got fantastic results from India and rice. It's really amazing. There's a bacterial pathogen in rice that's really a big problem, and there's no other treatment besides ascarosides, which seem to work extremely well. Okay, so these were just two examples. And they were both based on one single type of compound that we identified. There's many more compound leads that my lab works on. For example, these compounds here that we recently identified, they have, we believe, great potential in treating metabolic disease. They're compounds, again, that are derived from the natural environment, in this case of all vertebrates. This is something that humans make. So one less unknown compound in humans, also made in mouse and many other animals that we've looked at. So we believe that there are thousands of such opportunity in the hidden metabolome of plants, of animals, microbes, nematodes, and really understanding what these compounds do, especially in the interaction of the different species, will help us to develop new approaches to agriculture that are a lot more sustainable, that don't rely on killing things. And the same is likely true for drug development, where we can develop molecules that are based on things that naturally occur in the human body. So I've had a great team of researchers work with me over the couple of years. Here's the list of lab members. And of course, our work is super collaborative. So I really depend on collaboration with faculty here at BTI, for example, Dan Classic, but also with other faculty at Cornell, at Weill Cornell, New York City, and many other places all over the planet. Thank you very much. So we get that question so much, right? And there's all these other compounds that are, for example, salicylic acid mimics, right, that increase the disease resistance, and they all come with a penalty. We haven't found the penalty yet. So yield is up. So in every single successful trial, we have a yield increase. So what also happens is that it has a little bit of a growth stimulant effect. So if you go into these rice fields, you can see, oh yeah, those were treated with the ascaricide because they're greener, they're growing more. So there may be a multi-generational effect where it gets detrimental, right? We don't know that yet. But at least, as far as we can tell in the field, there's no penalty. What's the question over there? Yeah, I think. Yes.
If you examine the range of pests like Phytophthora, fungus, you have some sense of how this glycoside can this glycoside protect plants from other pests,
It protects generally broadly. The efficiency, it's broad protection, right? So in wheat or in soybean or in other plants that we've tested, we usually tested several different pathogens. Let's say a bacterial one, a fungal one, an oomycete one. And usually we see protection for all of them, although the extent to which we see protection varies a bit. Right? So sometimes in some species, protection against bacterial pathogens is really good, for example, in rice. And then in case of soybean, protection against the oomycete pathogen is really good. And perhaps protection against fungal pathogen is not as good. So, but this really relies on the classical priming pathway. So usually there's a broad.
So now, can you do this with one treatment?
One or two treatments very early on at a rate of 25 milligrams per acre. Okay, 25 milligrams per acre. I'm saying this to this audience, I feel embarrassed because I think nobody believes this. And also people in the field don't. So what we do is we mix we sell a premix, diluted solution, where then the farmer can add a whole liter or gallon, right, to their tank. So they see something substantial is actually added.
You know, we have several tree diseases like blights and elm disease, chestnut disease. Have you thought about testing this so you can inoculate your young trees?
Yeah, chestnut. So I have a plot of land in Italy, and there's many old chestnut trees that are, kind of on the verge of coming. It's a very interesting question. So we've tried it in apple orchards on Seneca Lake near Geneva, and we don't have yield data there, but visually, in terms of leaf drop for apple, for example, the effect is very significant. So we've been looking at more and more different crops, but of course, in the field, there's a lot of variability, and for the company, it's important to have robust data, right? So we have to have field trials in many different locations for any one crop that we engage with. But we have tons of compound available now. This molecule looks pretty complicated, but we can now make it at a 100 kilogram scale. So we've also developed a lot of chemistry there. Yes?
So it's a naturally occurring compound.
Yes.
That anybody could extract. You can't patent it, therefore. Commercialized.
Oh no, of course it's patented. It's patented. If you discover a new natural product, you can patent its use for certain purposes. So many drugs, right, they are patented, although they are of bacterial origin or they are of plant origin and so on. Right? So it's really the connection of discovering a new compound with an unanticipated use. And that's important for the company, of course, right? That we wouldn't be able to attract much funding probably without a good patent portfolio. Yeah.
So this ascaricide comes from the nematodes. Are there any effects on the nematodes that are in the soil where you plant?
Yeah, so what I forgot to explain is that these sclerocytes, they're made by parasitic nematodes, soil nematodes, and so on, and they are their language. They're pheromones that facilitate all kinds of signaling between different life stages of the nematode, between different individuals of the nematode. There's many different variants of the structure. And of course, we may be messing with that signaling network by treating the field. But the amounts that we treat with are very small. And also we can show that within 2 days, the compound is absolutely undetectable anywhere on the field. So it gets degraded very quickly. So we believe that really the impact on the ecology of the field is really minimal.